Hello and welcome to day one of EULAR News here at the EULAR 2020 Virtual e-Congress. We may not be together this year, but the program is very packed and we hope to cover some of the content over the next 3 days here on EULAR News. My name is Mrinalini Day and I'm an academic clinical fellow and junior doctor in rheumatology working in Liverpool in the UK. And I'm joined today and for the next 3 days by my 4 fellow reporters, the first of whom is Eduardo.
Hi, I'm Edoardo Prediletto and I'm a Versus Arthritis PhD student. I work in the Experimental Medicine and Rheumatology Department of Professor Fitzalice at the William Harvey Research Institute in London. And I'm going to cover mostly the part about translational medicine and basic science. And back to Sebastian, my colleague.
Hi, I'm Sebastián Rodríguez García. I'm a consultant rheumatologist. I'm currently working on La Princesa University Hospital in Madrid. And my work is currently focused on rheumatoid arthritis. Now I will introduce you to Fabian Proft.
Thank you, Sebastian. My name is Fabian Proft from the Charité University Hospital in Berlin, where I'm heading the clinical trials unit, with a strong focus on axial spondyloarthritis and psoriatic arthritis. And now I would like to hand over to my colleague, Mikhail.
Yeah, hello everyone. I'm Mikhail Protopopov from Charité Medical University, Berlin as well. And I'm working at the same department as Fabian does. So we are colleagues. And my scientific interests are also spondyloarthritis and psoriatic arthritis. And now I'm passing the mic back to you, Mrinalini.
Thank you very much, Mikhail. So today was day one of the EULAR 2020 e-Congress, which means that we had the opening plenary session today, which, Mikhail, I believe you are going to cover some highlights from. So what were your highlights from this session?
Yeah, indeed, Mrinalini, I had an opportunity to check the abstracts of the open plenary session. This is the session that publishes the outstanding abstracts that might be interesting to a wide society of the rheumatology. And there were indeed very interesting abstracts included in the session. For example, there was an abstract showing non-inferiority of upadacitinib versus adalimumab in patients with active psoriasis arthritis, presented by the EULAR president, Jan McInnes. There was a very nice report from China regarding the gut microbiota and its role as a trigger for the high-risk individuals for rheumatoid arthritis. But I would like to focus on 2 presentations that I personally find very interesting. First one is an analysis coming from Sweden, is a big retrospective study where they analyzed the prevalence of the anterior uveitis episodes in patients with axial SPA treated with different biologics. It is now well known that anti-TNF medications are very effective to control the uveitis in patients with axial SPA, but it is also known that in comparison to other drugs, etanercept seems to be less potential, less effective. And those data published by a Swedish group confirmed this. Another drug that was analyzed within the study was secukinumab. And although the data are hard to interpret due to channeling bias and due to selection bias, secukinumab was mainly used as the 2nd line or 3rd line or later line treatment. It seems to be, after adjusting for those differences, that the prevalence of the uveitis is not significantly higher in the patients treated with the secukinumab compared to the anti-TNF medications.
And that's very interesting, Mikhail. Can you just tell us a bit more about the prevalence of first episode of uveitis in this cohort?
Yeah, this is a very interesting question because the prevalence of the first episode of the uveitis in the patients treated with different biologics was very low and was not exceeding 1%. So regardless of the drug that was used. And the second study that I would like to draw your attention to is the data of a enhanced study showing the influence of the serum uric acid level on mortality. The authors really found that the mortality was U-shaped in this case, meaning that the low uric acid levels were associated with an increased mortality, especially in males, as well as high uric acid levels were also associated with the increased mortality. So I believe those two abstracts are really interesting for the wider audience and the session in general is really worth visiting.
Thank you very much, Mikhail, for going through that with us. So one of the highlights for me personally when I go to the EULAR Congress is to meet like-minded young rheumatologists, particularly other members of EMUNET, and to go to the sessions which they put on for us on various aspects of methodologies and research questions which are relevant to young rheumatologists. So Sebastian attended the Young Rheumatologist session today. Can you tell us about your highlights from the session, please, Sebastian?
Thank you, Mrinalini. Every year, EMUNET's working group members are given the chance to send proposal for next year's the Young Rheumatology session. Well, last year I proposed this about outcome measures in rheumatology because I believe it is a topic that remains unknown for some part of our community. And I also believe it should be well understood since it has an important impact in daily practice decisions. In this session, first, Dr. Sofia Ramiro gave us an overview on the subject and introduced the outcome measure properties, that is, validity, reliability, and sensitivity to change, and feasibility. And then she commented on the OMERACT filter and the cause-mean approach that is mainly used for patient-reported outcomes. And also, after talking about these properties, she reminded us about the EULAR outcome measure library, which we can browse and search for outcomes in different languages, and also order by disease whenever we are trying to initiate some research. And after getting an idea of these outcome properties, we also invited Dr. Aletaja, who presented a practical example on how to apply this principle and how they worked for develop the Simplified Activity Index, the SDI, that is widely known for people mostly in the rheumatoid arthritis field. Then, after explaining how they got this index, they also went for a critical appraisal of this index and how the clinical disease activity index appeared, this CDAI. And finally, Dr. Castrejón introduced the impacts of the patient perspective on these outcome measures and how tools such as RAPID-3 or the multidimensional health assessment questionnaire can also be of help in daily practice when making clinical decisions. And that way we got a broad overview about how we are measuring things in rheumatology nowadays.
That sounds really interesting and really useful sessions, Sebastian. Were there any oral presentations in this particular presentation session?
Yeah, there were 2 oral presentations, and I would like to highlight one of them on the development and validation of an alternative ASTES for its use for research purposes when patient global assessment isn't available. Briefly, the authors combined longitudinal data from 4 cohorts and 2 randomized control trials of axial spondyloarthritis, and randomly split the observations into a development cohort and a validation cohort, and considered BASDAI as the total score and single or combined BASDAI questions as to replace the PGA when there was not there. So in the development cohort, conversion factors for each PGA substitute were defined by general estimating equations, and a validation was performed in the respective cohorts, assessing the already mentioned outcome measure properties. So after comparing the different combinations that they had, they conclude that an alternative ASTAS using BASDAI total score as PGA replacement was the best approach for its calculation in cohorts when actually the PGA is missing. So those were the facts about these sessions that I wanted to highlight for today's EULAR news. So I hope you find it interesting.
So, Mikhail, I believe this session you attended was actually called Artificial Intelligence and Machine Learning, which sounds very exciting. Was there any artificial intelligence that particularly caught your eye in this session?
Yeah, thank you, Bernalini. Indeed, I had an opportunity of visiting or checking up the open plenary session, which combines the abstracts that are of high interest to the wide society of the rheumatology. And there were really indeed wonderful works there presented. For example, the work on the efficacy and safety of upadacitinib in patients with psoriatic arthritis, showing that this drug was not inferior to adalimumab, and this report was presented by Professor Jan McInnes. And the Chinese team was presenting a very nice data on the potential influence of the gut microbiota on the development of the RA in patients with the high risk for developing RA. But my first highlight would be the study that was presented by the Swedish group. The second oral presentation that I was going to highlight is literally future coming to your office. The group of the Danish scientists actually validated and showed how good the convolutional neural networks are able to grade the ultrasound sacroiliitis, and the ultrasound was also performed by a robot. So human being was not pretty much involved in the clinical care at this point. The ultrasound was really performed by a robotic device, and the data were analyzed by 3 connected convolutional neural networks, which were able to differentiate between no power Doppler signal and some power Doppler signal with a surprisingly high specificity and sensitivity. So if we are speaking about the modern or future technologies, this is really looks like it.
Thank you, Mikhail. That sounds really fascinating. I'll have to catch up on that session later on, I think. But now we'll move to something a bit different. So Eduardo actually attended one of the translational science sessions today, specifically on detection of type I interferon. And he's going to tell us a little bit more about that. And I was actually looking forward to this, particularly because of a landmark paper that came out earlier this year on the use of anifrolumab in SLE, which I believe is one of the abstracts he's going to cover for us, as well as a few other things. Over to you, Eduardo.
Indeed, Miralini. As a basic scientist, I'm always very into what regards translating the work we do on the benches to the patient beds. And so I've been following several interesting sessions, and all of them were amazing. And this is not flattery, but I'm very happy to highlight 2 in particular in the detection of type I interferon bridges into clinical application session. So the first talk I would like to mention is from Dr. Arman Howe of the Charity University Hospital in Berlin, Germany. And what they have done is to assess STAT1 and STAT3 expression and phosphorylation at baseline and after interferon type 1 and 2 stimulation. So basically what they have done is to check it on B-cell subpopulation of SLE patient, and then they compared it to other autoimmune diseases and healthy control also related all the experiments to the disease activity. So the results showed that the SLE patients showed substantially higher STAT1, but not p-STAT in B and T cell subset. And what is also very interesting is that increased STAT1 expression in B cell subset is correlated significantly with SLDI and CD169 on monocyte, which is a typical interferon 1 marker.
And what about the other cells, Eduardo? Did they also look at the expression of the STAT1 on the other cells?
Thank you, Mikhail. That's actually a very good point. So it is important to add that STAT1 activation in plasmablast was interferon alpha dependent, while on the other side, the monocyte exhibit a dependency on interferon gamma. So in conclusion, I can say that, in my opinion, this talk is relevant because it's a nice lab work of flow cytometry, and also they are actually investigating the YAG-STAT inhibition, which is a very attractive therapeutic possibility for SLE. So, and the second talk I wanted to mention, as Mrinalini introduced before, regard a very hot topic for the SLA community, which has been followed with great interest as a new hope for patient treatments. So I'm actually talking about the work of Professor Eric Moran of the Monash University of Melbourne, that together with partner and colleagues, they have actually recently published on the New England Journal of Medicine. So in a nutshell, the talk covered mostly the outcomes of 2 phase 3 studies, the TULIP-1 and TULIP-2, where they evaluated the efficacy and safety of a monoclonal antibody drug, which is named anafrilumab, which bind the interferon 1, the type 1 interferon receptor. So just a brief description of the talk. During these studies, Moran and colleagues, they randomly assigned 362 patients with SLE to receive either 300 milligrams of the drug or placebo every 4 weeks for a total of 48 weeks. And then among the participants, 180 were treated with the drug, while the other obviously received the placebo. So the complete primary endpoint was a BICLA response at week 52, and this included a reduction of moderate to severe baseline disease activity, as well as no worsening of any of the 9 organ system in the BILAG index. So the results showed during the presentation are quite encouraging, I think, since according to the researcher, 47.8% of the participants in the anafrilumab group achieved a BICLA response compared to the 31.5% of those treated with placebo.
So, Eduardo, I would like to know, did they stratify the patients through the interferon gene signature?
Thank you, Sebastian. I'm very glad that you mentioned this. It's a very good question. So what is obviously more interesting, since you're dealing with interferon here, and it also is kind of intuitive, is that for a second outcome, the patients have been also stratified between an high or a low interferon gene signature. And among those, the results are even more interesting because 48% of the patients treated with anafrilumab demonstrated a BICLA response compared with 30.7% in the placebo group. So overall, I think that my comment on this is, as also mentioned by Dr. Moran, is that this is the only second drug to have a positive phase 3 trial in SLE in the last 60 years. And as biological uptake in SLE management remains low, patients are still mostly treated with steroids or immunosuppressive drugs. So little has really changed since the 50s, but a plethora of monoclonal antibodies and small molecules are on clinical trials at the moment for SLE. And you can just think about rituximab, baricitinib, or the IL-12, IL-23 pathway, ustekinumab. I mean, I honestly think that this is a breakthrough finding supporting the efficacy of this drug. And not just for that, but also for us researchers, is basically validating the type 1 interferon pathway in the pathogenesis of SLE, which honestly, I think really opens thousands of doors in finding the origin of the disease.
Yes, so thank you very much, Eduardo. And speaking of SLE, one of the mainstay of treatments that we have is, of course, hydroxychloroquine, which has been in the news a lot lately due to its, well, it was a possible role in the treatment of COVID-19. As of recording this news show today, the arm testing hydroxychloroquine has been pulled from the WHO trial. But as I said, it's a drug that's well loved and used by rheumatologists for SLE and rheumatoid arthritis. So I'm going to be covering an abstract from the session on biologic DMARDs in rheumatoid arthritis, which looked at the possible association between hydroxychloroquine use and the development of heart failure in patients with rheumatoid arthritis. And this was done on a cohort from Minnesota in the USA. And the researchers actually had a split cohort of 143 patients who went on to develop heart failure and 143 patients who didn't, all of whom had rheumatoid arthritis according to the ACR classification criteria. Those who developed heart failure were classified according to the Framingham classification criteria. And approximately half of both cohorts, so around 70 patients in each cohort, had been given hydroxychloroquine. So the results of this study showed that hydroxychloroquine was not actually associated at the doses that we use with the development of heart failure in patients with rheumatoid arthritis. And there was no statistically significant association between cumulative dosing of hydroxychloroquine with heart failure.
Brinalini, and did the researchers look at the other cardiovascular outcomes, such as, for example, the prevalence of the myocardial infarction or overall cardiovascular mortality?
Thank you, Mikhail. That's a very good question. And actually, those outcomes were not presented in this particular presentation, but of course, they are very important outcomes. And similarly, it wasn't clear whether or not the relative effect of other cardiovascular comorbidities, such as diabetes, hypertension, had any significant effect on the end outcomes. As we know, in rheumatoid arthritis, the patients are more at risk of developing cardiovascular complications, of course. And it's important to say, however, the study was strengthened by facts such as they had good information on hydroxychloroquine, which they were able to report. And of course, they had a matched cohort of patients who didn't develop heart failure, which is always important in these studies. So that was my highlight from that particular session. Do join us again tomorrow. So in tomorrow's EULAR News Show, we will be covering a wide variety of topics, including highlights from sessions on spondyloarthritis, rheumatoid arthritis, and some of the patient sessions as well. Thank you for joining us today on day one of EULAR News. We hope you enjoyed our highlights. Do go across to the eCongress portal and check out some of the sessions for yourself if you've not already done so. See you again here tomorrow. Bye.
Bye-bye.
Bye.